I want to share with you some personal background because it helps put my own views about evolution into perspective. I am a physiologist. That means I'm interested in how living systems work. In particular, I'm a physiological ecologist. That means I'm interested in how living systems fit into the environments they inhabit. My research career has been shaped around a simple question. What does the science of physiology have to say about the science of evolution? Adaptation has always been at the center of that question. In exploring this question, I've worked with all kinds of creatures, ranging from alligators to incubated birds' eggs, embryos in birds' eggs really, and each has illuminated some aspect of adaptation. What really challenged my thinking, however, has been this. This is a mound built by a species of termites that inhabit southern Africa. The mound that you see here is built by a colony of 1 to 2 million termites that live underneath it. The termites don't inhabit this mound, oddly. They live in a nest situated about a meter below the ground. What the mound is instead is a wind-driven lung. It's literally an organ of physiology for the colony, built by the many termites that inhabit the colony. So this is an organ of physiology, a lung built de novo out of sand, clay, and termite spit. Like our own internal organs, like our lungs, the mound lung is structured in a way that it functions well. Furthermore, the mound can change shape depending upon circumstances. If there's a tree nearby blocking the wind, for example, the mound will adjust its shape to better capture the energy in wind. In short, this is a spectacular example of adaptation, and it's adaptation operating at a scale much different from how adaptation operates in organisms. This was a real challenge to my thinking about adaptation, and it forced me to go right back to first principles. And it's out of that challenge that heterodox thoughts about evolution have come. To lay the foundation for what's to come, we'll look at what probably is the primitive physiological function, managing the cell's volume. The cell is the fundamental organizational unit of all life. It consists of a highly specialized functional interior separated from an exterior environment by a cell membrane. The cell membrane is not a barrier. It's a permeable boundary between two environments, internal and external, and materials can flow back and forth across it. This includes water, which can flow into or out of the cell through specialized pores embedded in the membrane. Cell volume is controlled by these water movements. If water flows in faster than it flows out, the cell will swell. If water flows out faster than it flows in, the cell shrinks. For the cell to sustain its volume, water flow in has to equal water flow out. This is an important point. There is always flow. A steady cell volume does not mean that there is no flow of water. It means the two flows are equal. The flows of water across the membrane can be controlled by the concentrations of two electrolytes: potassium, symbolized with an uppercase K, and sodium, symbolized by Na. Both electrolytes are ions with a positive charge. There's something peculiar here. The interior of the cell typically has a high concentration of potassium, expressed here in large letters, and a low concentration of sodium, expressed in small letters. Outside the cell, sodium is at high concentration and potassium is at low concentration. In other words, the membrane spans significant concentration differences of these two electrolytes. These concentration differences don't just happen, they are built by the membrane. As it does for water, the membrane has built into it specialized pores for potassium and for sodium. Whenever there's a concentration difference across the membrane, both electrolytes will flow down their respective concentration gradients. This happens all by itself. And this means there will be a continuous flow of potassium ions from the interior of the cell to the outside, And conversely, there will be a continuous inward flow of sodium ions from the exterior to the interior of the cell. This is getting a little complicated, but hold on, because it's going to get just a bit more complicated. If the electrolytes flow down their concentration gradients, the concentration difference would eventually disappear. That the concentration gradients nevertheless persist means that the loss of potassium from the cell and the inflow of sodium are offset by some type of pump. Even a leaky bucket can be kept full, for example, as long as you pump water in as fast as it leaks out. The persistent concentration gradients of sodium and potassium work the same way. The membrane pump uses energy to do work, pumping potassium into the cell as fast as it leaks out. At the same time, it pumps sodium out of the cell, again as fast as it leaks in. Okay, now remember that the flow of water across the membrane depends upon the internal concentrations of these electrolytes. Therefore, the volume of the cell can be adjusted by altering the flows, emphasis flows, of electrolytes across the cell membrane. These can then adjust the flows, again, emphasis flows of water across the membrane. One more piece. To regulate volume, the cell has to know what its volume is. That comes from another component in the cell membrane, which senses the stretch in the membrane. It's like a balloon. The more a balloon is inflated, the more the balloon's rubber membrane is stretched. And it's the same with the membrane cell of the cell. If the cell's volume increases, the cell membrane stretches. If the cell's volume decreases, the membrane relaxes. Membrane stretch sensor informs the cell what's happening. The stretch sensor, in turn, uses that information to adjust the porosity of the potassium pores, or the pores for sodium, or for water. The information can also adjust the activity of the pump. If the cell swells, this means that water is flowing in faster than it's leaving. The swelling is detected by the membrane stretch sensor, and all these components working together can be adjusted to oppose the swelling by reducing the influx of water and increasing water loss. If the cell shrinks, this means that water is flowing out faster than it's flowing in. Same principle. The stretch sensor can detect this and increase the flow of water in and reduce the loss of water until they come back into balance. Phew, that was complicated. So far, this is just mechanism, just physiology, how adaptation can work, but it hasn't told us much about what adaptation is. Adaptation, remember, is the tendency to aptitude. In the context of our cell, this means that the cell will keep its volume steady, even if the environmental conditions change. That's adaptation. Say, for example, external sodium concentrations drop, as they would if the environment was suddenly inundated with fresh water. This will affect the fluxes of ions across the membrane and the water fluxes. Specifically, water will begin to flow faster into the cell, causing it to swell. To keep cell volume steady, the water fluxes must again be brought into balance. So, for example, the sodium channels might open, the potassium channels might close, the water channels might also restrict the flow of water, and the pump might be made to work more intensely. In this way, the various controllers of flow of electrolytes and water across the membrane are adjusted so that the volume of the cell is maintained, even as the external environment changes. That is adaptation. Okay, that was a dive into the weeds. We'll be glad we took the plunge, however, because it opens the door to a more general theory of adaptation. That general theory will be the jumping off point for the deeper dive into evolution that's to come. Let's start with a real cell. This is a common type of organism that lives in ponds, Paramecium bursaria. It's green because it's photosynthetic, like a green plant. Unlike a plant, this cell swims around its habitat using numerous cilia, which you can see is the beating fringe around the cell. We're going to recast adaptation, and life for that matter, as a problem of thermodynamics. This cell represents an internal environment that is separated from an external environment by the cell membrane. The interior of the cell looks very complex. Indeed it is. But let's not get lost in that weed patch for now. The cell's environment is characterized by a high degree of orderliness. You can tell that just by looking at the cell, but let's reframe this. The highly ordered contents of this cell are made up of elements like carbon, hydrogen, oxygen, nitrogen, and others. The cell exists in this form because these elements have been organized into the cell's highly ordered internal environment. They form proteins, carbohydrates, nucleic acids, complex lipids, all those things you learned about in general biology. All are highly ordered molecules, and they are organized in a highly ordered way. The external environment consists of the same materials, namely carbon, hydrogen, oxygen, nitrogen, etc. Outside the cell, these form molecules like oxygen gas, water, carbon dioxide, and so forth. All are much simpler molecules. They are less ordered. So what distinguishes the cell from its surroundings is its highly specified orderliness. There's just one problem. Orderliness is inherently unstable. If left to itself, it will automatically degrade to disorder. This is why dead cells decay. They are reverting to a natural state of disorder and releasing heat in the process. Heat is the ultimate form of disorder. Yet the living cell persists. The question is, how does the cell persist even as it's continually degrading to disorder? The answer is that physical work has to be done to take this disorderly matter and convert it into the orderliness of the cell. Heat is also produced as a byproduct of this transaction, but so is orderliness. And this is the fundamental thermodynamics of life and adaptation. Persistence of the cell means doing work to create orderliness out of disorder as rapidly as the cell spontaneously degrades to disorder. That work continually cycles matter through unstable states of order into stable states of disorder. That's the foundation upon which a general theory of adaptation can be built. Stop and rewind if you have to. This is a very important point. There's just one more little bit of vocabulary housekeeping we need to do. Orderliness and disorderliness are representations of a fundamental thermodynamic principle known as entropy. The concept of entropy can be a little tricky to keep straight. It's often misunderstood. You can blame the physicists who defined it. But here goes. Entropy is a measure of disorder. High entropy means high disorderliness. By contrast, orderliness means low entropy. Thus, the cell is really a pool of low entropy that exists in a sea of high entropy. Just keep that straight and you'll avoid a lot of confusion. Okay, back to adaptation. A general theory of adaptation will be a thermodynamic theory, plus something else. It's the something else that brings in the heterodoxy. You'll see what I mean momentarily. Central to our general theory of adaptation is a so-called adaptive boundary. We'll just represent this as a line dividing an exterior environment from an interior environment. Adaptive boundaries take space and partition it. In the case of a single cell, the adaptive boundary is the cell membrane. But as we shall see, adaptive boundaries can encompass far more. Spanning the adaptive boundary is a gradient in entropy. External to the cell, entropy is high, and disorder is likewise high. Inside the boundary, the environment is more orderly. Low entropy prevails there. Maintaining the entropy gradient is the array of membrane components we've already introduced. There's an engine that uses energy to do work. In the general sense, this is work to reduce entropy. There's also a set of selective channels through which the unstable low entropy of the interior degrades to disorderliness. Together, these compose what we might call an orderliness engine. We could also describe it as an entropy reduction engine. The channels themselves could be described as entropy production engines. An adaptive boundary also has mechanisms for control. These devices can control both the entropy reduction engine and the entropy production channels. The controller can make the pump work harder, for example, or it can control the rate at which entropy is produced. Here's where things get interesting. Up to now, all this is standard physiology and mechanism. The important question about adaptation is what ultimately controls all this, and here's where the heterodoxy starts. An adaptive boundary also includes what we might call a cognitive engine. This consists of some means to compare some attribute of the external environment to the internal environment. And depending upon the outcome of this comparison, this can modify the controller, shown here as an octagon, and in turn the orderliness engine. Adaptation involves keeping the specified orderliness of the internal environment fixed, even in the face of a variable external environment. Imagine, for example, that entropy in the exterior environment goes up. That could come, say, from an increase of temperature. The entropy gradient between interior and exterior will therefore increase, and rate of degradation to disorder will also increase. Sustaining the cell's internal orderliness will mean increasing the entropy reduction rate. Adaptation involves sensing of this change by the cognitive engine and a modification of the orderliness engine to sustain the specified orderliness of the cell's internal environment. This is our general model of adaptation. We'll be expanding on this in subsequent episodes, but for now, there's one very important point to take away. Adaptation involves manipulation of flows of energy and materials across an adaptive boundary. The mechanisms for doing this are the bread and butter of physiology, but there's more than simple mechanism at work here. What makes this an adaptive boundary is the presence of the cognitive engine. Adaptation means having some means of sensing the state of the external environment, assessing how this might affect the state of the internal environment, and then taking steps to set the mechanisms for adaptation into motion. In short, adaptation is cognition. The mechanisms are servants to the cognition, and you cannot explain adaptation without cognition. And here's the really big point. You can't explain evolution without cognition either.